Hi and welcome again. My name is Gonzalo Artiach and I'm a healthcare equity analyst here at ABG. Our next company is Q-linea, a diagnostics company focused on infectious diseases. And with us, we have here today the CEO, Jonas Jarvius. Jonas, the floor is yours.
Thank you very much. Also, my apologies for not being able to attend the Haymarket today. I would really love to, but we have so many meetings coming up in the afternoon, so we'll have to do it over link. But happy to be here. Thank you very much for that. I think we can move into the next slide immediately, where we have our disclaimer slide, of course, in case me or my colleague Anders Lundin would make any forward-looking statements. When you have read that now, I suggest we move in and look at some of the highlights of Q-linea. So we can take the next slide here. Thank you. So we are based in Uppsala, so not too far from Stockholm. We are focused at developing disruptive and improved infectious disease diagnostics tools. ASTar, our main product, as you can see to the right, is CE marked. We as a company, our CE-IVD, are approved, meaning that we can continue to develop products under the new regulation. So we are a little bit over 117 employees and consultants currently in the company. We are currently in an active phase of revising our go-to-market strategy. I'll come back to that a little bit in my presentation. But if you look at ASTar, really, we have now, as I said, been on the European market for more or less a year. We're ramping up that market. We have filed for the FDA for our access to the US market. And we also received the breakthrough device designation by the FDA, which is really, of course, a very high quality stamp and shows the importance of our technology for US customers. And as I mentioned, we are now fully IVDR certified. One of very few companies actually in Europe that has actually met that timeline. And we also are in discussions with our next product in development, but today's focus will really be on ASTar. So if we take a look at the next slide. When we think about the problems we want to solve, well, bloodstream infections, where sepsis is the most severe condition, is our number one problem for blood analysis. Sepsis is a huge problem. Every third person that will die in our hospital will die from sepsis. And even if you combine our most common cancer forms, you still see a higher death rate in sepsis. So this is not a minor problem, it's an actually huge problem. And diagnostic really comes into the picture where we can see today that we start therapy, but the current diagnostic workflow will take too long. And that actually means that a lot of patients will have died, passed away until you receive the results. So providing faster active results in sepsis care is of key importance. If we take a look at the next slide. So as I mentioned, of course, sepsis being the first application, but as a part of what we do, we of course want to contribute to minimize antimicrobial resistant development. This is something that has escalated clearly over the last, I would say, 10 to 20 years. But we see that common bacteria become more and more resistant, become more and more difficult to treat. And it has been presented as the biggest threat to mankind. So this is no small feat. And of course, Q-linea will not solve this piece of the puzzle, but to provide accurate diagnostic indications, what drug to use, can really help in reducing AMR. And I think this is very important. If we then move into the next slide, of course, what we do is to serve a market. And this is a market with a very big unmet need. So overall, when we look at it, we see a multi-billion-dollar addressable market, more or less $1 billion addressable market only for blood. But ASTar was designed really from day one to be a platform. So yes, the first application is for blood analysis, but we have already demonstrated use for isolate, which is a huge market, some 90 million tests, Europe and US. We have also demonstrated urinary tract test analysis. So we really see it as a platform that can grow with the increasing demands in the market and can, of course, attract newer tests to that platform. So if we take the next slide, we have seen that it's a huge market. And really, what's the problem we want to address? Well, you can see it on this picture here. The top part really illustrates the traditional diagnostic workflow for septic patients. And without going into any details, you can see it's a multi-day, multi-step process. But the big problem is that it takes typically 3 or 4 days before you get the results. And what is the result then? Yes, it is what antibiotic should I use to treat this patient's infection with? And at this time, around 20% of the patients you started on diagnostic therapy have died. They've passed away. So this is what we want to help with ASTar, is to provide that actionable results up to 48 hours faster. And not only doing that, we also want to ease on the work burden for the people in the lab. So it takes just around 2 minutes to start the test. And if you compare that with the top part where you have multiple interactions with multiple instruments over a several day period. So it's ease of use for the lab and of course a better outcome for the patients. So if we then take a look at what the meaning of this could actually be. If we see the next slide coming up, we can look at, these are our 3 independent health economic studies that has shown the benefit of 24-hour faster diagnostic. And from left to right, you can see that a dramatic reduction in mortality because the treatment then actually works. Of course, less pressure for these opportunistic super infections, where you can see a lot of that in the lab, and also common infections, because you don't have to treat broad. And I think for the hospital, the biggest driver perhaps is that this study in particular shown that you could save on average 2 days in the intensive care beds. And 2 days in the most expensive beds, where you have also typically quite a few of them, is a huge cost saving. So I think all this is good support, but everyone, of course, want to know how do ASTar perform in this. So we are now coming very close to start our first multicentric study in the southern part of Europe. So of course, that's going to be interesting to be able to announce start of the study and then of course coming back with the results during next year. But that's the overall effects. If we take the next slide, of course, I mean, a lot of you have probably have a friend, relative that has been developing sepsis. So this is just one patient case. It's a 64-year-old gentleman who had surgery. He went on empiric therapy. He developed sepsis after that surgery treatment, and he was set on pip/tazo. So that's a combinatorial, very common drug combination for septic patients. And what you can see on the lower part is all the timestamps, what happened in this in his case. You could see in the morning you had a gram stain indicating bacteria in the blood. You can indicate what type of bacteria. It was a gram negative. And you could see that early the next day, you had a positive sample. So you could then immediately, of course, load it into ASTar. And if you take the next slide, you can also see that ASTar could, during the same day that the sample came in the morning, it could provide results on 13 more antibiotics. It could also tell you that pip/tazo was not an ideal therapy for this patient and could suggest alternative treatment regimes. The hospital immediately shifted therapy and the patient had a very positive outcome. The hospital themselves made the assessment that if they had not had the possibility to act so fast, it's a very high likelihood that this man would have died the next day. So this case, it was 18 hours faster, but it truly demonstrates that from a personal plane, it's not just health economic. These are individuals that can benefit from faster diagnostics. So if we take the next slide, we see here that who are then the companies trying to address this market? Well, the way we look at it, we basically see 4 companies in the market that either work with fully automated solutions or semi-automated solution. And semi-automated means that you have to do a little bit more hands-on time in the lab. But what we now have seen ASTar performing, we can see that it's the only platform that combines the full automation, very simple to use, with random access, with a high throughput, and also combining fastidious and non-fastidious bacteria. And we also do that with the broadest platform of antibiotics and concentration ranges of all companies in the field. So I think that we have developed a very good and very strong system, and it's going to be interesting now to see us coming more closely to market and see how that develops. If we take a look at the next slide, of course, these are the 4 pillars that we designed ASTar around. We did it together with hospitals on many years, and it was really ease of use, fast, comprehensive, and accurate. And I think the comprehensive part here is really a game changer. And what we mean with comprehensive is really that if you have a broad panel, the likelihood that you can actually act upon the result or a patient sample that you load in ASTar is high if you have a broad panel. So if we look at these 4 pillars and then see how has it actually turned out? Well, we can take a look at the next slide. So this is just what we now see customers running ASTar or in trials. And we can see that super simple to use. We can really have our staff doing other things. And also, I think the most impressive part here is summary from 2 studies. And the lowest number we have seen so far, of course, these are not many hospitals, but in 95.4% of everything that you loaded on ASTar, you could act upon that. That's a very high number. So really, I mean, what we set out to do in the comprehensiveness of the panel really starts to demonstrate its value. If we then look at the next slide, this is the ease of use. I mean, we designed it to be less than 2 minutes, very simple interaction. You're guided by the graphical user interface on ASTar, and it's seamlessly integrated in the current lab flow. You don't really have to do anything else. You just spend a couple of minutes on ASTar, and then you can leave it. So very simple to use. If you take the next slide, we can also see that high throughput is, of course, important. We designed ASTar for medium-sized and up hospitals, so typically serving 500 beds and more. And this, of course, means that you can, in the morning, load a lot of samples. If you're a daytime lab, take care of the peak in the morning. And if you're sort of a larger lab, you can have a high sustained throughput with random access. And we've also designed our consumables to be able to address fully automated mode, which we do now for sepsis, but then also be able to run semi-automated for, let's say, less time-critical samples. And then you only load the disk. So this is under development, but it also means that the same system can address full automation at one price point, semi-automation at a very different price point. So even there, we have built the system to be able to have a broad capabilities for sample types and workflows. But then take a look at the next slide. I think this is, of course, the performance data of ASTar. And as you can see to the left, this is just data from our clinical study. We are well above guidelines in Europe and US. And you can see to the right that our ranges, I mean, in what concentration range can you determine the susceptibility of bacteria? Because some case you have a native wild type, so a friendly E. coli, then you should treat that with a low concentration. But you could also have a super resistant, multi-drug resistant E. coli or ESBL bacteria, which require very higher concentration. It could still be the same drug, but if you have this broad concentration range, you can adjust therapy, PK/PD measurements adjustment for, let's say, the friendly E. coli that still can have a patient being killed in sepsis, and the most severe ones. And this is really one of our strengths in the panel. If we then move on to the next slide, we can also see that we have been building Q-linea for a long period of time. And we have been doing that together with customers. And we have really spent time in the hospital trying to identify what are the problems in the workflow. And we have now focused on ASTar, as you can see to your right, for rapid AST. But we have also identified that the transportation of these samples from the patient to the lab takes a long time, over 12 hours sometime. And the goal is to be beneath 2 hours, at least in the US. So that's where Podler come in, and that's really bringing point of care capabilities for sepsis diagnostics to the bedside. You actually utilize the workflow in transportation for the diagnostics. So it doesn't really change workflow for the lab or for the staff, but you can use time instead of waste time. We also have capabilities in the middle here with our multiplexed ID technology. This is nothing we are developing right now. We have demonstrated an absolute, I would say, world-class performance in highly multiplexed ID analysis direct from venous blood, so not requiring a blood culture. And this could be the next disruptive steps in 7 to 10 years, and we have the technology to take us there. But to the right, we have AST, and of course, ASTar being our lead product, what we are really promoting now. But we have also demonstrated that we can use the same technology as we have in ASTar for rapid point-of-care diagnostics. So we have done that for urinary tract samples within 45 minutes, providing a guideline on what to treat. So our sort of technology in the company have always been focused on developing in the workflow for the lab and for the patients, and it's covered by 24 patent families. So it's a broad coverage. I think if you take the next slide, we'll see a highlight on ASTar. And this is, of course, our key focus. That's where we now put a lot of our efforts. If we take the next slide, of course, this means that we want to contribute to the world. And I said I'm going to talk a little bit our go-to-market strategy, and I will do that to this slide. As some of you have heard, maybe all of you, is what we ended our partnership with Thermo Fisher Scientific just a couple of months ago. And what we are doing right now is, of course, implementing our new go-to-market strategy. And that will be a mix. We do want to focus to have Q-linea incorporated in the US. We have a foothold in the US. We already have employees over the last year to support in clinical studies. We want to extend that reach because US is really our key market. But of course, we are not going to look at sort of taking on the entire world. We are now in discussions with distributors, and we are looking at discussions for high-level, sort of broader distributors, but also more regional distributors. And really, our goal is to find the best solution, either for a country or for a region. And we are now moving and making good progress in those discussions. What we also have learned is that our last year in Europe, we saw that it's a very long sales cycle. Rapid AST is very new in Europe compared to the US, where they actually have seen Rapid AST, and it's a much more mature market, which I would like to see us coming to that market and see benefit from that traction. But Europe is not there yet. We haven't seen Rapid AST. I mean, all the companies I basically presented are just approaching the European market. And we see that customers are asking a lot of questions. Can I try Astra before I buy it? And of course, we have seen a number of evaluations, all positive. So we have received very good feedback. But then we are facing a long tender process. And of course, this is difficult because you, as well as I, would like to see much better traction in sales. And I think it will come because the key core is something that really has been positively received. But we also see, as I mentioned, we have a number of colleagues in the field that also want to do Rapid AST. And we see that there are a couple of labs that want to do an evaluation comparing our system with a DRAST system, for instance, or even with Celluix Reveal system. So we are now also coming up in the late stage of having independent evaluation, which is, of course, important to cross-compare this platform and see what are the different pros and cons with them. So I think that we are, of course, doing a lot of work now implementing new go-to-market strategy. We're also having a lot of visits together with Thermo Fisher in the handover from customers or evaluation sites or just sort of sales prospects. So we probably need a couple of more months before we can really demonstrate and show you that new path forward. But I think the key here is that we have built a very strong system. We see that we have a huge unmet need, very few companies today addressing that need. So I think that if we look a little bit long term, I think we can see a very good development of our company and together with you, of course. So with that, I will actually pause and open up for some questions, giving a little bit more time for questions today, and I hope that's fine. Thank you very much.
Thank you very much, Jonas, for your presentation. And as you say, now we have time for a few questions. And keeping the discussion from the last part, I would like to start with a question on the termination of the deal with Thermo Fisher. As you have mentioned before, now you have two possible scenarios that are open in front of you. Either you continue on your own, your commercial growth, or you move with a new partner. And in the scenario where you move forward alone, I was wondering how difficult will it be to address new areas, for example, the US, as you were mentioning before. And you have said that you already have some employees there, but how much would you have to grow your infrastructure there, and how realistic is to target this market alone?
So first of all, sorry if I made that impression. We are not going to target the market alone. We are going to work with distributors in different geographies and different areas. So that's going to be an absolute requirement, and that's going to happen. And we are in those discussions. But what I think is a key for us now is also to extend our own commercial organization, primarily to the US, because it's such an important market. So we want to have our own foothold, but of course then also work together with distributors, because US is much more consolidated than Europe, for sure. It's one country, but it's a large country. So I think that's the strategy. I think the good part is also when you look at US, there's very good statistics on what hospitals are running a lot of blood culture samples, because there's statistics on everything. We also know who are the sweet spot for us. And if you look at sort of the top 100 first customers, we already know who they are. So even a sort of a moderate sales force of around 15 people can really start making a difference to those key accounts. But it will be a journey together with distributors. And as I said, we're looking at both sort of more specialists at some countries, for instance, but also regions. And we're also talking to much larger companies that really would like to see the entire US or entire world, for instance. And that's really where we are now trying to mitigate. So the answer is we will not go alone, but we have learned a lot during the time with Thermo Fisher because it was a partnership. So we had a mirror organization. We participated a lot in these discussions with customers. So in that sense, we have a running start. We just need to extend that reach.
Thank you. One of the questions that we have gotten from investors is, what is the impact in your P&L of the termination of this deal? Could we expect to see a negative impact in terms of instruments that Thermo Fisher had and were already perceived as sales coming back to you, so returned instruments in the next quarter?
I wouldn't say that you can see that as a negative impact for sure, but of course what we're also sad is that during this transition period, we will need to see, we can't just take over a customer contract, for instance. That needs to be done in a negotiation. And so far these negotiations are going well, but it needs to move in the right phase. So I don't see that type of impact. But of course, what's the difference now is that historically, we reported a sale when Thermo Fisher bought an instrument because they were our sort of customer. Now, of course, all sales will be reported when we sell it to an end customer, alternatively to a distributor. That could be similar in the size. So we might see a mix of sort of registration of sales over the next year.
Okay, thank you very much. I have a next question on Podler. I mean, this device fits quite nicely in your whole story. But so far, we don't know that much about the strategy behind the commercialization of this product. I was wondering if you could give us a little bit of color on this, and also what are the leads that you're planning to target? Is it hospitals, or would it be more like microbiology labs?
Right. So the first part is, of course, as we announced, that we enter discussions with Podler, and we are really looking at the go-to-market strategy with Podler, partnership or ourselves. Currently, of course, we are really revising that strategy because we know that we will now focus some part of our internal sales force. And of course, it's really a nice combination of Podler and Astar. You could think about bundle deals in the future. So we're currently revising really and say, what's the best way to move forward now? Is it to keep Podler and Astar completely separate in our discussions, as we have done in the past? Because, of course, we had Thermo Fisher for Astar. So we're now sort of having a revised go-to-market strategy for Podler in that sense. If you look at who will be our customers, I would say it's still the microbiology labs, because they will really see the benefits of the Podler in their workflow, meaning that instead of wasting time in the transport that's currently happening, they can just have a continuous diagnostic workflow. The true benefits of that will, of course, be the patients that could save huge amount of time of using transportation time for diagnostic workflow. But also for the lab, we see in the US a lot of hub and spoke arrangements, and you can think about a scenario where you have a number of Podlers coming into the central lab. Since Podler is fully autonomous, if you have a positive growth in one of the blood cultures, that will indicate that. So immediately when it arrives to the lab, you can do segmentation and prioritization, say we have 3 samples that are already positive. Let's put them directly into the workflow. The others can just be on the bench, incubate it, and you can really take them sort of when you have more time. So I think it will really help the hospitals prioritize the patient with the sort of the positive cultures.
Thank you. And one last question on Podler. Different regions have different healthcare structures. For example, the US is more centralized than Europe. Could we expect a larger and faster uptake of Podler in the US compared to Europe?
Yes, I mean, the way we look at it today, to be honest, is I think we're going to see a faster uptake for both Astar and Podler in the US. And also in the US, we tend to look a little bit more from a business sense, to be honest. So you can actually see that saving time in transport, increasing diagnostic sort of outcome is something that the hospital will benefit from. So we see that we have the same sort of transportation issues in Europe, but I would say that Europe is a highly likelihood to adopt that. We also see a lot of hub and spoke key hospitals in the US, so a large region. And of course, their Podler will have an even higher value than in some parts in Europe.
Jonas, I think we're running out of time. Thank you very much for Jonas' presentation. And
thank you very much as well, and nice to contribute, although not at the Haymarket. Sorry about that.